Welcome back to the Practical Jobs to be Done Deep Dive. Today we're really getting into something that I think a lot of people are misunderstanding.
Massively misunderstanding.
Right. We are drilling down into the bedrock of what is arguably the most hyped, well-funded, and frankly confusing sector in tech right now.
Yeah.
We're talking about the collision, and I'm using that word deliberately, of artificial intelligence and biology.
Collision is definitely the right word. It's violent, it's messy, and there are a lot of casualties that people just aren't seeing yet.
See, that's why I wanted you here for this deep dive. Because I walk into this topic all wide-eyed. I mean, I'm looking at the miracle. If you read the headlines or track the venture capital flowing into this space, the narrative is basically that we've already won.
Yeah, the victory lap is in full swing.
Exactly. You have AlphaFold solving protein structures. You have these massive GPU clusters. I mean, NVIDIA is practically printing money from biotech demand alone. And we have these new Agentic AI models that supposedly generate novel drug molecules in weeks. Weeks. It used to take years.
It did take years. And to be fair, for the generation part, you're absolutely right. It's almost instantaneous now.
Right. So the story we're being sold, and that you as a listener have probably heard, is this complete paradigm shift. We're moving from biology as a discovery science, guys in white coats holding up test tubes, hoping something reacts, to biology as an engineering discipline. We write the code solves the biology, we cure cancer, we cure Alzheimer's, and we live forever.
It's the singularity pitch.
It really feels like we're on the cusp of it.
It feels that way, yeah. It's a very seductive narrative. It's elegant. It appeals to our desire for control, especially over our own biology. But, and this is a massive but, which we're going to spend this deep dive unpacking, there's a huge difference between generating a solution on a computer and validating a solution in a human body.
The gap between the screen and the patient.
Exactly. Right now, the industry is confusing the map for the territory.
Okay, let's get into the source material that's sparked this, because we're basing this conversation on a really brutal piece of strategic analysis. It's a document titled The In Silico Illusion.
It is a sobering read.
Very. It centers on a deconstruction of a company they call Cell Type, but realistically, it's tearing apart the entire thesis of the agentic drug discovery industry.
It's a necessary takedown, honestly, because while the tech press is throwing a parade for the speed of discovery, the actual pharmaceutical reality is incredibly stubborn.
Lay the groundwork for us here. What is the core conflict that this document exposes?
The core conflict is essentially a war between two metrics. On one side, you have the Silicon Valley metric,
Which is speed.
Right, speed to clinic. Move fast, break things, iterate, get the code running, get the molecule designed as fast as humanly or artificially possible. On the other side, you have the pharmaceutical reality metric, which is clinical survival.
And I'm guessing those two don't map onto each other very well?
Not only do they not correlate, they might actually be negatively correlated right now.
Wait, naked?
Yeah. Just because you get to the FDA toll booth faster doesn't mean you actually have the right ticket to get through the gate.
Okay, I want to roadmap this for you listening, because we are going to go really deep today. We're going to start with this fallacy of speed, why going fast is actually breaking the business model. Then we're going to get into the financial physics.
The math in this section is wild.
It is. I saw the breakdown of compute cost versus the cost of human failure, and my jaw actually dropped. It leads into something they call the ID10T index.
We will definitely spend some time on the ID10T index.
After that, we'll look at the biological barrier, why the code fails when it hits actual wetware. And finally, the report doesn't just complain. It outlines 3 very distinct pathways out of this mess. 3 ways to actually fix the industry.
Lateral, sustaining, and disruptive.
Exactly. So let's start with section 1, the fallacy of speed. Because as an outsider, speed just seems inherently good.
It usually is.
Right. If I can use an AI agent to discover a drug candidate in 3 weeks versus 36 months using a team of human chemists, how's that not a massive win? You just saved 35 months of overhead.
In almost any other industry, you would be 100% right. If you're building a SaaS product, speed is everything. Launch the beta, find the bugs, push a patch. If you're building hardware or cars, manufacturing speed improves your margins directly.
So why is biology different?
Because drug discovery is unique.
Yeah.
The feedback loop is not instant.
Like if my code has a bug, the compiler throws an error in 2 seconds.
Precisely. In drug discovery, you generate a molecule today. You don't actually know if it works in a human being for 5 to 10 years.
5 to 10 years just to find out if your code works.
Exactly.
Yeah.
So imagine you speed up the front end. You're racing a Ferrari to a toll booth. You get there in record time. You feel like a genius. But the toll booth is just the start of clinical trials, and the gate is down. Speeding up the drive to the toll booth didn't give you the money to open the gate. You just arrived at the traffic jam faster than everyone else.
And this traffic jam, this is what people call the Valley of Death, right?
Yes. Specifically, Phase II efficacy testing. We have to be crystal clear about this because it's the biggest misconception in the space. The bottleneck in medicine is not finding molecules.
It's not.
No. We have physical and digital libraries of billions of molecules. Finding a shape that fits a target is not the hard part anymore. The bottleneck is knowing which of those billions of molecules will actually work in a chaotic, messy human body without killing the patient.
The report mentions a statistic that really kind of halted me. It says the 90% clinical failure rate has barely budged.
That is the headline nobody in Silicon Valley wants to print. Despite all the billions of dollars, despite AlphaFold, despite the massive server farms of H100s and H200s, the failure rate for drugs entering clinical trials is effectively the exact same as it was 10 years ago.
That's crazy.
Roughly 90% of what we put into human trials still fails.
So all this AI firepower, all this compute,
Yeah.
it's not actually making us smarter.
No, it's just making us faster at guessing.
Faster at guessing.
Yeah. We are essentially speed running the tutorial level. The AI is generating what the report calls sophisticated false positives, molecules that look absolutely perfect to the algorithm, they look perfect to the human chemist reviewing them, but when you put them inside a living, breathing system, they crash and burn.
But hold on, let me play the optimist here for a second. Even if the failure rate stays at 90%, if I can fail faster and drastically cheaper, isn't that still a massive business advantage?
The shots on goal argument.
Yeah. If I can run 1,000 cheap AI experiments in the time it used to take to run one expensive human one, eventually the law of large numbers says I'll win, right?
This is where we have to talk about the financial physics. This is the numerator versus denominator section of the document, and it's brilliant because it exposes how Silicon Valley logic literally traps the biotech startup.
Okay, let's break this down. The efficiency delta. Walk me through the math.
Let's look at the old way first, the human cost. The numerator. Traditional drug discovery is incredibly labor-intensive. You need PhD-level scientists. In a biotech hub like Boston or San Francisco, the benchmark loaded rate for that kind of talent is roughly $78 an hour.
Loaded meaning salary, benefits, lab space overhead, all of that.
Right. And you don't just need one scientist. You need teams of 5, 10 of them working full-time for years. The total preclinical cost just to get one drug candidate ready for an FDA application ranges somewhere between $300 million and $600 million.
That is a staggering amount of money just to reach the starting line of human testing.
It is. It's wildly inefficient. So now look at the denominator, the compute cost. The physics floor for this new agentic AI industry is the GPU cluster. The report specifically benchmarks the NVIDIA H200 cluster. They use AWS P5e 0.48x large instances for the math.
That is incredibly specific.
It has to be, so we can price it out accurately. In early 2026, the spot price for that kind of massive global compute power is roughly $39.80 per hour.
Okay, let me just pause there. $78 an hour for one human chemist versus $39.80 an hour for a supercomputer that can simulate, what? Millions of chemical interactions.
Tens of millions. In an hour, it can do the data processing work of 1,000 chemists. So you, the visionary tech founder, look at that spread. $78 versus $40. And you say,
we just reduced the cost of discovery to zero.
Exactly. You declare victory.
It seems so obvious. You trade the expensive, slow wetware, the humans, for the cheap, fast hardware. Your margins go through the roof. Your throughput goes infinite.
But here is the trap. And if you're listening, this is the most critical point of the entire financial argument. Listen to this very closely. If you spend $39.80 to generate a drug candidate using AI, and that candidate looks good in the software, what do you do next?
You test it. You put it in a clinical trial.
You start a Phase II clinical trial on a human. Do you have any idea how much a Phase II trial costs?
I'm going to guess it's a bit more than $40.
The average is about $150 million. And remember what we said about the failure rate?
It's 90%.
Right. So if your ultra-cheap compute generates a sophisticated false positive, a drug that looks flawless in code but fails the moment it hits a human liver, you haven't actually saved any money. You have just accidentally triggered a $150 million loss.
Oh, man. Put that way, the math changes completely.
The true cost of that AI computation wasn't $39.80. The true cost was $150,039.80.
That is a terrifying number.
It destroys companies. It is currently destroying companies. If you use AI to accelerate the generation of bad ideas, you are just accelerating your own bankruptcy. You burn cash faster than any biotech in history. The savings you bragged about on the front end are literally a rounding error compared to the catastrophic cost of the failure on the back end.
So basically, unless the AI actually improves the ultimate success rate, unless it lowers that 90% human trial failure rate, the speed is a fatal liability.
Exactly. Speed without accuracy in biotech is suicide, and that beautifully leads us to the ID10T index.
The ID10T index. When I first read this, I was hoping it meant what I thought it meant.
Well, in traditional IT support, an ID10T error is a user error, because if you write it out, it spells idiot.
Right.
But in the strategic report, they use it as a cheeky way to calculate institutional waste in pharma.
How do they calculate an ID10T score for a modern company?
You take the commercial price of drug development, that infamous $2.6 billion average cost to get one successful drug to market, and you divide it by the theoretical physics floor of digital generation we just talked about.
And the massive gap between those two numbers is the waste.
A gap is the waste. And the report argues that the real ID10T factor here is that we have the roles completely reversed. We are paying premium human salaries, that's $78 an hour, to do robotic tasks.
Like what?
Data entry, pipetting liquids into plates, basic molecular optimization. We are using humans as manual laborers.
And the robots.
We're using cheap compute to generate these false positives that we blindly treat as gospel. We are trusting the code too much and utilizing the human intellect too poorly. We use PhDs as data clerks, and we use GPUs as a magic 8-ball.
That is a scathing indictment of the whole system.
And the house always wins.
Right, and in this metaphor, the house is biology itself.
Exactly.
So let's talk about the house. Let's get into the biological barrier.
Right.
Why is the code failing? Because logically, if the math is perfect, if the simulation is running on the most advanced NVIDIA clusters with literally all the public biomedical data in the world, why aren't these AI-generated drugs working?
This is what the paper calls the in silico illusion. And it comes down to a fundamental misunderstanding by computer scientists entering the biospace. We have to remember, biology is not a deterministic software environment.
It's not code.
It is not code. In software, if you write a script that says, if A, then B, the computer will always execute B, it is deterministic. Biology is a chaotic, stochastic, wet physical system.
It's messy.
It's incredibly messy. The report highlights off-target effects as the primary failure mode for these AI models.
Walk us through off-target effects. How does that happen?
Okay, let's use an analogy. Imagine your AI is tasked with designing a key. The drug is the key, and it needs to fit a very specific lock, let's say, a malformed protein on the surface of a cancer cell. The simulation runs millions of variations, and it finally says, here's the key, perfect fit, maximum binding affinity.
Success. We print the key, we cure the cancer.
In the computer simulation, yes. But then you put that physical key into a human bloodstream. The key floats around, it eventually finds the cancer cell, it locks in, great. But it turns out there is a receptor on the tissue of your heart valve that happens to look kind of like the lock on the cancer cell.
Oh, and the key fits that too.
It fits just enough, and suddenly, while your brilliant AI drug is successfully shrinking the tumor, it is also stopping the patient's heart.
And the simulation just totally missed that.
The simulation didn't see it coming because it wasn't looking for it. It was completely optimized for the target lock. It wasn't simulating the entire chaotic universe of the rest of the human body.
It's the problem of unknown unknowns.
Precisely. You can't compute what you haven't modeled. And then you have metabolic breakdown.
The liver.
The liver.
The report calls the liver the bouncer, which I thought was a great way to put it.
It is the toughest bouncer in the biological club. The liver's entire evolutionary job is to look at absolutely anything you swallow and ask one question. Is this food or is this poison? If it looks foreign, the liver destroys it.
It doesn't care that you just spent $10 million designing this perfect molecule.
The liver does not care about your seed round.
Yeah.
You can make an AI molecule that perfectly destroys cancer in a glass test tube. Amazing.
Right.
But when a human swallows it, the liver breaks it down into inactive garbage before it ever reaches the tumor. Or worse.
Worse.
The liver tries to break it down, and in the process, metabolizes it into something highly toxic that destroys the liver itself.
And we can't just simulate the liver to prevent that.
Yeah. Not accurately yet. The human liver is a chemical factory running thousands of parallel enzymatic processes simultaneously. We literally do not have the source code for it. This is the black box problem. For many complex diseases like Alzheimer's or autoimmune disorders, we don't even fully understand the mechanism of the disease itself.
Right. We don't know the rules of the game we're trying to hack.
Exactly. So how can an AI simulate a cure for a disease when the scientists themselves don't understand the physics of the disease? It can't. It's guessing based on incomplete data. And this perfectly explains the Phase II chasm.
The chasm. Break the numbers down for that.
Currently, AI-generated drugs are hitting about an 80 to 90% success rate in Phase I clinical trials.
Which, again, if I'm a retail investor looking at a press release, 90% success sounds incredible. I'm popping champagne.
And that's exactly the trap they use to raise their Series B, because Phase I is just safety testing.
It's just asking, does this immediately kill a healthy volunteer?
Basically, yes, is this cyanide. And usually with modern tools, we're pretty good at designing things that aren't instant poison.
Right.
But Phase II is efficacy testing. That's where you ask, does this actually cure the disease in a sick patient? And that is where the success numbers crash down to 40% or lower.
Wow.
And that 40% is completely indistinguishable from the old school non-AI human baseline.
So the AI isn't improving the only part of the process that actually matters.
No, it's just getting us to the failure point faster. And a lot of this comes back to the mouse.
The mice. I feel like every single time we do a deep dive into biotech, we end up talking about mice.
We have to, because the entire industry is violently addicted to what the report calls the false proxy.
It's true, though. We've cured cancer in mice a million times over, haven't we?
We have the healthiest, most cancer-free mice in the history of the universe. If you happen to be a mouse with a glioblastoma, you are in luck. We have incredible health care for you. But a mouse is not a human being.
It sounds so obvious when you just say it out loud.
It is obvious. But the entire pharmaceutical pipeline is built on this legacy assumption that if a drug works in a mouse model, it might work in a human. But the genetics are fundamentally different. The metabolism is different. A mouse's immune system operates drastically differently than ours.
So why do we still use them? Why hasn't the industry moved on?
Because the FDA accepts the data.
Wow. So it's just a compliance thing.
It's a box you can check.
Yeah.
It is regulatory compliance masquerading as scientific truth. And here is the absolute kicker for the AI side of things. Guess what data these massive AI models are being trained on?
Oh no, don't tell me.
We are feeding the AI millions and millions of data points derived from mouse experiments over the last 50 years.
So we're building these trillion-parameter super intelligent systems that are absolute world experts at curing mice.
We are mathematically optimizing for the wrong species.
Garbage in, garbage out.
Mouse in, mouse out. We are generating sophisticated false positives because our foundational ground truth data is actually a biological lie.
Okay. Wow. That is heavy. We're, halfway through this deep dive, and I feel like we've just methodically dismantled the entire tech bio hype cycle. The speed is a financial trap, the math actively works against the business model, and the biology is simply too complex for the current code. Honestly, if I'm listening to this and I'm running a biotech startup right now, I'm probably curled into a ball under my desk.
Well, don't curl into a ball just yet. The analysis in this document isn't purely doom and gloom. It actually proposes concrete solutions.
Right, the pathways. The report outlines 3 distinct ways to pivot out of this disaster. Pathway A, Pathway B, and Pathway C.
Lateral, sustaining, and disruptive.
Let's start with Pathway A. They call it lateral persona expansion.
Which is consultants speak for the pivot.
The pivot. This is basically just admitting defeat on human biology, right?
I wouldn't call it defeat. I'd call it intense pragmatism. It asks a very basic business question. If our AI engine is legitimately amazing at designing chemistry, but human biology is just too chaotic and hard, where else can we sell chemistry?
So take the sports car off the dirt track and put it on a paved road.
Exactly. You take the exact same AI engine, the exact same code base, the same H200 GPUs, but you stop looking at human diseases. Instead, you point it at agrochemicals, advanced materials, and industrial chemicals.
Why is that a better business?
Regulatory arbitrage.
Okay, that sounds like pure finance speak.
Yeah.
What does that actually mean for the scientist?
It's the art of avoiding the FDA. An FDA trial for a human drug takes 10 years and costs $2.6 billion. But getting a registration with the EPA or the USDA for a new crop pesticide or a new biodegradable plastic.
How long does that take?
2 to 4 years total.
That is a massive difference in time and capital.
Massive. And think about the physics of the problem. A polymer, a piece of industrial plastic, doesn't have an immune system.
Right.
It doesn't have a liver. It doesn't have a chaotic, unpredictable response to a new molecule.
So the simulation actually maps to reality.
The simulation matches reality nearly 100%. If your AI simulates a plastic that is highly heat resistant and then you synthesize it in a lab, it will be heat resistant. There is no biological black box to ruin the experiment. The sim-to-real gap just disappears.
That makes total sense. You're basically playing the game on a lower difficulty setting where the engine actually works.
Exactly. And don't forget veterinary medicine.
Oh, right. To treating pets and livestock.
Yes. In veterinary medicine, the animal model is the patient.
Wait. Oh, that's brilliant.
Right. If your AI designs a drug and you cure cancer in a dog in the laboratory, you have literally just cured the end patient. There is no translation gap where you have to cross your fingers and wonder if the dog data will translate to a human. You are directly selling the drug to the dog owner.
So the false proxy problem vanishes entirely.
Completely vanishes. And the commercial logic here is incredibly sound. The report talks about LTV over CAC lifetime value versus customer acquisition cost.
Right, standard sauce metrics.
Yeah, but applied to bio. It is demonstrably better to take $20 million in realized actual revenue in 18 months from an industrial chemical company than to hold a lottery ticket for a 12% chance of making $1 billion in 10 years from pharma.
Especially if you're a startup trying to survive what the report calls the VC crunch.
Exactly. They predict a massive funding crunch hitting biotech in 2026. The tourists are leaving the space. If you want your company to survive, you need cash flow now, not clinical promises for a decade from now. Pathway A funds your survival.
Okay, so Pathway A is essentially run away from human pharma, go make money where the physics are easier.
Okay.
Just smart business.
Yeah.
But as a society, we still want to cure human disease, right?
Yeah.
We don't want all our brightest AI minds just making better dog treats and farm pesticide.
We definitely need to cure humans, and that is where Pathway B comes in, sustaining innovation. This is for the companies that want to stay in the human pharma fight, but actually fix the broken process.
So how do we fix the main pipeline without abandoning it?
By fundamentally changing the goal of the AI. Currently, most of these AI startups are selling novelty. They walk into a pharma company and say, look at this incredibly cool new molecule. Nobody has ever seen this shape before. It's totally novel.
But novelty doesn't equal safety.
No. Novelty often equals toxicity. We need to stop selling novelty and start selling viability. Because the chief scientific officer of a big pharma company, the person actually writing the checks, they do not care about your algorithm. They don't care that you use agentic workflows or whatever buzzword you have.
What do they care about?
Risk mitigation. They care about not getting fired because a $150 million Phase II trial blew up on their watch.
Okay, so how does the AI actually help with risk mitigation?
We have to shift the AI's objective function. We shift from optimizing for binding affinity, how well the drug sticks to the target, to optimizing for AIDMET.
AIDMET. I've seen this acronym a lot. We need to break this down because it seems to be the bread and butter of Pathway B.
It stands for absorption, distribution, metabolism, excretion, and toxicity.
The 5 pillars of existence for a drug.
It sounds super basic, but let's just look at the A, absorption. 40% of new chemical entities generated today fail because they do not dissolve. They are what chemists refer to as brick dust.
Brick dust, literally.
Basically, you make a brilliant AI-designed pill, the patient swallows it, and it passes right through their digestive tract like a stone. It never actually gets absorbed into the bloodstream because it's completely insoluble.
That seems like something we should probably know before we spend $100 million testing it.
You would think. But traditionally, the pipeline checks that stuff much later. They get totally hyper-focused on the lock and key fit first. Pathway B says, use the AI to predict the mundane failures like solubility or liver toxicity before the drug is ever physically synthesized in a lab.
So instead of using the AI to find the magic needle in the haystack, you use the AI to digitally burn down all the hay that doesn't matter.
Exactly. Filter out the trash first. If the AI predicts the molecule is going to be brick dust, delete the file. Do not spend $1 synthesizing it. This is where the document explicitly references the Musk loop.
Yes, I love that part. The Elon Musk engineering philosophy applied to biology.
It applies perfectly. Step 1 of his loop, make the requirements less dumb. Step 2, delete the part.
Delete the part. How does that translate to wet lab biology?
It means if a digital test or a physical assay does not directly correlate to human survival, stop doing it. Delete the step from your pipeline. Don't run an assay just because the industry has always run it.
Strip the process down to only the absolute bare essentials that predict human success.
Right, and then you integrate what's left with CROs, contract research organizations. They call this hybrid validation.
How does that actually work in practice?
You connect your AI directly to the CRO's lab via an API.
Software directly talking to a remote lab.
Yes. The AI designs the experiment digitally. It sends an automated order via API to a robotic lab facility somewhere else. The robotic arms execute the physical test. The physical data flows directly back into the AI model.
So human chemists are totally out of the loop.
Mostly, yes. It massively speeds up the iteration cycle. Instead of a scientist writing an email with a spreadsheet, sending it to a lab, and waiting 3 weeks for a PDF report, the API orders the test instantly and parses the data instantly.
That sounds incredibly efficient.
Yeah.
But you're still renting the lab. Right. And you're still relying on standard biological tests.
You are, and that is the inherent weakness of Pathway B. You are still renting someone else's lab. You are still just improving the old legacy system. You've made yourself a vastly better, faster cog, but you are still a cog in the same old broken machine.
Which naturally leads us to the big one, Pathway C, the disruption. The report calls this network inversion, and honestly, it sounds like a complete burn-the-boat strategy.
It is the ultimate solution, and frankly, it might be the only one that truly solves the in silico illusion.
So the premise here is, if the data is the bottleneck, meaning if the historic mouse data is bad, and if the public internet data is commoditized, then we need to literally own the physical generation of the data.
Yes. You cannot just scrape the internet for the data you need to cure cancer. You cannot scrape a biological trade secret.
What is the secret? What's the data that's missing?
Negative data.
Negative data. But usually, companies want to hide their failures, don't they?
And that is exactly the problem. Pharma companies bury their negative data. They don't publish peer-reviewed papers saying, hey, everyone, look at this billion-dollar mistake we made and why it poisoned the liver. They hide their shame to protect their stock price.
Nobody else learns from it.
Exactly. But if you are training a foundational AI model, knowing precisely what does not work is arguably more important than knowing what does work. The model needs to know the exact physical boundaries of failure. Pathway C says, stop being a software company entirely. Build a massive robotic wet lab.
They call this the CapEx inversion. Spend the capital expenditure, build the hardware, build the factory.
Build the factory, but not just any factory. You build a closed-loop robotic lab that operates almost entirely on organoids.
Organoids. Okay, this is the sci-fi part of the deep dive. Explain organoids to me like I'm 5 years old.
Okay, imagine a standard computer microchip. But instead of silicon transistors carrying electricity, it has tiny microscopic channels filled with fluid. And inside those fluid channels, we grow actual living human cells.
Real human cells.
Real human liver cells. Real human heart cells. Lung tissue. We call it human on a chip.
So no more mice.
No more mice. Or at least a very, very drastic reduction in mouse testing. You completely bypass the false proxy of the mouse and replace it with a miniaturized human model.
So if the AI's drug is toxic to a human liver, you see it happen on the chip before it ever touches a real patient.
Immediately. Imagine the workflow of a company running Pathway C. The AI agent designs a novel molecule at 2:00 a.m. It sends the digital instructions to a robot down the hall.
The robot wakes up.
The robot physically synthesizes that drug at 2:05 a.m. It injects it into a human liver organoid chip at 2:30 a.m. The microscopic sensors on the chip detect cellular distress or toxicity. That negative feedback data flows directly back to the AI model by 6:00 a.m.
That is insanely fast. And it's real human biological data.
It is immediate human relevant feedback. And here is the true business mode. That data, that specific negative toxicity data from the human organoid does not exist anywhere on the public internet. OpenAI doesn't have it. Google DeepMind doesn't have it. Only you have it because your physical robots generated it that morning in your lab.
Wow, that transforms the identity of the company completely. You aren't just an AI software tool anymore.
You become a translational engine. You aren't selling software subscriptions. You are selling biological truth. You are selling the absolute certainty that this molecule will not kill a human being in Phase II trials.
That is incredibly powerful. It feels like the difference between being a graphic designer who draws a picture of a bridge and being the actual construction company that builds it.
or the difference between drawing a map and paving the road. Now, Pathway C is violently expensive. It requires massive capex. You have to build the physical lab, you have to hire robotics engineers, you have to hire wet lab biologists. But if you pull it off, it builds an absolute monopoly on biological truth.
So if we look at the landscape right now, entering 2026, companies basically have a choice to make.
They do. They can be a feature that's Pathway A or Pathway B. You are useful, you are potentially profitable, but ultimately you are just a replaceable component in someone else's machine. Or you can be a platform Pathway C, where you own the entire physical and digital loop.
The reality check here, and what this report makes so clear, is that the future of this industry isn't just about an AI that thinks faster than a human.
No. It's about AI that is deeply grounded in physical reality. The era of computer says yes being enough to raise $1 billion is over. We are entering the era of computer says yes and our robot physically proved it on human tissue.
If this actually succeeds, if Pathway C really works at scale, what does that do to the field of biology as a whole?
This is the final provocation of the report, and it's profound. If we succeed at this, biology truly stops being a discovery science. It stops being about guys in lab coats accidentally stumbling upon penicillin in a moldy petri dish.
And it becomes...
What? It becomes a true engineering discipline. Predictable, repeatable, scalable. Just like building a suspension bridge or designing a silicon microchip. We finally take the chaos of life and subject it to the precision of engineering.
That is a staggering thought, turning the chaos of life into the precision of engineering.
It is the ultimate goal. But I will remind you, and everyone listening, until we actually bridge that Phase II gap, until we stop failing 90% of the time in human trials, it's all just a bunch of very expensive video games.
Extensive video games. I love it.
We have to physically earn the right to call biology engineering.
Well, that is a whole lot to chew on today. From the ID10T index to the false proxy of the mouse to growing human organs on microchips. The core message of this deep dive is loud and clear. Speed is fake. Survival is real.
Viability over novelty. Always.
So to everyone listening, I want to leave you with this final thought. Look at your own industry, whatever it is. Are you blindly optimizing for speed, or are you optimizing for true viability? Are you just figuring out how to get to the toll booth faster without actually having the ticket to get through the gate?
That's the question we all need to be asking.
Thanks for joining us on this Practical Jobs to be Done deep dive. We'll see you next time.